without further ado, I think it's time to introduce our second keynote speaker of the meeting. And she's going to close out this historic virtual meeting. And I just wanted to say that looking at the past keynote speakers that have spoken in person at Cold Spring Harbor, I'm shocked that she wasn't on the list already, based on her accomplishments, of course, in retrovirology, and the fact that I think I've been coming here for 18 years. I think I've seen her in the audience every year, including after she became a professor emerita. And I think she probably was going to the meeting when it was still known as the tumor virus meeting. So she's been a longstanding member in the retrovirus community. And I'm hoping she'll also give us some perspective about being one of the pioneer female scientists in the early days of retrovirology and molecular biology. Just a little bit about Anne, for those of you who don't know her. She started her graduate studies actually at Yale University shortly after women were first allowed to attend graduate school there, but her PhD advisor left to take a position across the country, and she didn't want to leave the New York area. So she transferred to NYU, where her thesis investigated how histones affect DNA and RNA synthesis in Jerry Hurwitz's laboratory. She then moved to Cold Spring Harbor to do her postdoctoral fellowship, and she and her family actually lived on campus in the Hooper House, which is a short distance away from Rice Auditorium, right down the road. And there, Anne studied lambda phage transcription in Alfred Hershey's laboratory. Then after she did her postdoc, she started her own lab at the Roche Institute of Molecular Biology, RIMB, starting at the equivalent of assistant professor, but then rising through the ranks over 18 years and eventually becoming a department head there. It was there that she switched from studying lambda phages to the molecular biology and biochemistry of ASV. And in 1987, she left RIMB and became the scientific director of the Institute for Cancer Research and senior vice president for basic sciences at the Fox Chase Cancer Center in Philadelphia. And there she continued to lead a well-funded laboratory for nearly 30 years. Her extensive retrovirology research includes work on reverse transcriptase and mechanisms of reverse transcription and recombination. She also worked on integrase and integration, proviral gene transcription and its implications in oncogenesis, and protease. And she helped write the first edition of Principles of Virology with our friend Vincent Racaniello, and she co-organized this meeting in 1996. She is also an elected fellow of the American Academy of Arts and Sciences, the American Academy of Microbiology, and the New York Academy of Sciences. But in addition to her vast research accomplishments, I wanted to note that she served on a number of high-profile committees. I'm just going to name a couple of them. She was the representative on the Committee of Genetic Experimentation called COGENE, which was an international council of scientists that advise international governments on recombinant DNA technologies. And many of the protocols that they developed were actually included in the Molecular Cloning, a laboratory manual by Maniatis et al. By Cold Spring Harbor Press that many of us use in our labs still today. And since 1983, she continues to serve on the New Jersey State Commission on Cancer Research, including a stint as the chair, which is an appointment by the New Jersey governor. And finally, she spent 11 years on the Defense Science Board to advise the US Secretary of Defense on scientific and technical matters. And she was awarded the Medal for Exceptional Public Service by the US Secretary of Defense in 2012. And as we mentioned, she recently published a book that's very exciting, and she'll provide that to many winners today. So I hope all of you in the audience will help me welcome Dr. Anne Skalka.
Okay. I'm having technical difficulties here.
Okay, you can just press slideshow tab and say start show.
I lost the screen. I lost,
You think? No, there we go.
Let's see. Do you have it?
Yep.
Oh, great. Screen sharing has failed to start. Please try again later.
It looks fine. It looks fine.
Okay.
Good.
All right, let me say okay. Okay. All right. So am I in and everyone's seeing that my slides? Great. So I'm really most honored to receive this invitation to talk about my career as a scientist and in particular, I suppose, as a woman in science. And as I look back on my research experience over the last 6 decades, egads, I was struck by how much our research styles and capabilities evolved as we went through one technical and cultural revolution after another. So I thought that I would focus particularly on some of these changes, especially in the beginning of my career. But first, for a short bio to set the stage. I was born in 1938, and these were tough times, just after the Great Depression and just before World War II. Not too unlike our current situation for some, I guess, except without the social isolation. My dad immigrated from Austria, and my mother was from Puerto Rico, a somewhat unusual combination, but not so much for New York City. So I grew up in a working class neighborhood and was a product of New York City public schools. My dad worked at the Charles Pfizer production plant, which was then in Brooklyn. And it was there that I had my first experiences in a lab, working as a research technician's assistant during my high school summer breaks. Upon graduation, this is 1955, few of my friends and none of the girls were headed for college. But I was fascinated by science and with the scholarship support, went on to major in biology at what was then a small liberal arts college, Adelphi College, on the South Shore of Long Island. I did some undergraduate research in herpetology at Adelphi with my major advisor. We even published 2 short papers on fossil turtles. However, a pivotal moment for me came in our biochemistry lab when we did an extraction of DNA. To provide some perspective now, this was 1957. The classical era of genetics had just ended. It was only 5 years since Hershey and Chase had convinced the scientific world that genes were made of DNA and not proteins, as was the common belief before then. And it was only 4 years after Watson and Crick. So I was fascinated by the realization that one could actually see and touch the stuff of genes. I became hooked on biochemistry and genetics, and then encouraged to apply for a graduate program. Well, on to graduate school. As Andrea mentioned, I started out in the microbiology department at Yale Medical School, which at the time was an all-male institution. They had just started to admit women, but only into the graduate school. And we were all housed together in a woman's dorm on the main campus. However, I decided to transfer to NYU Medical School after a year, when David Bonner, my mentor, moved to California. And I eventually, as Zandia said, that doing my thesis work with Jerry Hurwitz at NYU. Now, again, for perspective, this was the 1960s. The central dogma, DNA makes RNA and RNA makes protein, had just been established, and the genetic code was not yet deciphered. Arthur Kornberg had discovered a DNA polymerase in E. coli, but it was only the dawn of the golden era of molecular biology. Jerry Hurwitz was a young and ambitious nucleic acid biochemist who had just co-discovered the DNA-directed RNA polymerase in E. coli, and I was one of his very first graduate students. Now, Jerry was a really tough taskmaster, always working in the lab himself, constantly looking over our shoulders, even taking our lab notes home with him at night to review. The department included a number of female graduate students, however, and postdocs, but everyone was very competitive, and we all had to learn how to defend our ideas in a group of really sharp and critical professors and colleagues. Because eukaryotic chromosomes were known to contain proteins called histones, although they were not very well characterized at the time. The project that I was assigned was to determine if and when how histones or how they could affect the synthesis of RNA and DNA by the E. coli polymerases. Now, the tools of the trade were quite limited by today's standards. We had to make our own nucleic acid templates, enzymes, and labeled nucleotide substrates. No convenient supply houses or kits were available yet. We could determine the base compositions of our templates and products, but nucleic acid sequencing was not even a dream. Fortunately for me, histone preparations from various sources were provided by outside collaborators, so I didn't have to go there. Well, I quickly discovered that, as had others, that addition of positively charged histones to nucleic acid templates inhibited polymerization. But to determine if there was any specificity to this suppression, I used the only method then available called nearest neighbor analyses. Now, one would start by including a nucleotide triphosphate labeled with P32 in the alpha position, such as the GTP shown here at the left, as one of the substrates, and then treatment of the product with sodium hydroxide or an enzyme that cleaved in the 3' position would lead to the transfer of label to the adjacent nucleotide in RNA or DNA products of digestion. One then repeated the reaction using each labeled nucleotide separately. My results were included in Hurwitz's presentation at the Cold Spring Harbor Symposium in 1963 and in this 1966 JBC paper. Now, among our conclusions was the observation that the DNA in tissues from various organs of an animal did not vary in base composition and nearest neighbor frequencies, something that seems quite obvious now, but it was not at the time. I also found that in the presence of histones, transcription from complementary strands was not inhibited equally, and the changes in nearest neighbor frequencies varied, depending on what kind of histones were added. In our discussion section, we made the following somewhat prescient observation that copying of various DNAs is inhibited to different degrees by various histones, and that this presents an obvious temptation to speculate on their roles as regulators of gene function. However, we admitted that our meager understanding indicated a need for caution. Well, caution indeed. Of course, at the time, we had no knowledge of a nucleosome structure or even how histones interacted with the DNA. A few other important events occurred in my life at the beginning of that decade. In 1960, I was married to Rudy Skalka, a CPA who was then working in New York City for the accounting firm of Pete Morrick Mitchell, and who has been my anchor and support ever since. Well, our wedding had to take place during the Christmas break because taking time out of the lab for such frivolity was much frowned upon for students at the time. In 1962, I took the phage course at Cold Spring Harbor. It was there that I heard a talk by Alan Campbell, who had just reported his hypothesis of how bacteriophage lambda DNA integration might occur. Campbell proposed that this phage DNA, which was known to be a linear molecule, is converted into a circle after it's injected into the host cell. Cleavage of the circle at an internal site, an insertion of the DNA into the host chromosome at that site, could explain the puzzling differences between the genetic maps of phage DNA and the integrated prophage DNA, which were known to be circular permutations of one another. Well, I was fascinated by the potential dynamics of lambda DNA and in the way that genetics and biochemistry were used to deduce the mechanism. When it was time to decide where to go next, at my request, Hurwitz wrote to Al Hershey, the famous phage geneticist at Cold Spring Harbor. As Hershey replied in his letter that they certainly could use someone with my expertise, in 1964 I was off to Cold Spring Harbor, and there I became a member of the famous phage group, while my gracious and accommodating husband traveled every day on the Long Island Railroad to his office in New York City. At Cold Spring Harbor, we lived in the newly renovated apartments at Hooper House, shown at the top right, and I worked in a corner at the top floor of Demerit's lab for most of my time at Cold Spring Harbor. It was a huge change for me, from an ultra-controlling mentor to Al Hershey, who had a total hands-off policy. Everyone in the group was free to work on whatever we pleased, and we only got advice when we asked for it. The main focus of interest in the Hershey group at the time was trying to figure out the nature and functional relevance of the peculiarities and different features that had just been discovered in the various phage DNAs, as shown here in the middle. Hershey and his colleague, Betty Berge, had discovered that lambda phage DNA had sticky ends, which turned out to be short complementary single strands, which could explain how Campbell's circles form from linear DNA structures. The T4 phage DNA was a linear duplex. It never integrated into host DNA, but mysteriously exhibited a circular genetic map. And T5 was also a linear duplex, but it contained a nick in one strand. Well, I zeroed in on lambda, and a few new experimental tools had become available for my explorations at the time. Hershey had shown how linear DNAs could be broken into fragments of defined average length by controlled hydrodynamic shear in a Vertis homogenizer, no restriction enzymes yet. And together we showed that mercury added to cesium gradients made by centrifugation in a standard angle rotor would get excellent separation of the DNA fragments dependent on their GC content. Now, messenger RNA could be labeled by pulse chase experiments and purified by hybridization on crushed agar columns in which single strand DNA was embedded, called augured columns in those days. So messages isolated at different times following infection could be matched with DNA fragments. And of course, we had mature phage genetic systems with known mutants and so forth. So the box here shows a summary of my major findings. Lambda DNA was found to comprise 6 distinct segments ranging in GC content, shown at the top, from 37 to 57%. And the segments were found to be reasonably homogeneous internally, with borders between segments rather sharp. The genetic map also revealed a correspondence between the segments and the encoded functions. Late functions were encoded in the left, highest GC contents segment, and early functions on the right with lower GC. Regulatory functions were at the center with the lowest GC. It was just as if different functional chunks of genome had been acquired from different contributors and put together in a kind of patchwork, a notion that later proved to be extremely credible. Now, this was a very productive period for me with multiple publications documenting the summary that I just provided, but I put these up for another reason. You will note that Hershey's name is only on one of these publications, 1967 on the right. Furthermore, I am the sole author on two, one for a talk that I gave at the 1966 Cold Spring Harbor Symposium, and a second on a 1966 paper in PNAS. The one or two co-authors on other papers were either collaborators at other institutions, Butler and Eccles in 1967, or lab mates. Merv Smith in 1966, when we discovered that lambda and T5 DNA had made concatenamers in infected cells. And with Phyllis Baer in 69, when we mapped the origin of the then elusive lambda repressor. Well, it turns out that Hershey had a policy that his name would not go on a paper unless he had personally performed an experiment that was included. It was an extraordinary and generous policy for those of us in the group, although I can assure you, none of us failed to benefit from his very sage advice, and most importantly, his careful, sometimes even painful editing of every publication from our group. It was a productive time for me in another sense as well. Our daughter Jeannie was born in 1966, and in 1969, just as I was to leave Cold Spring Harbor, my handsome son Chris was born. Well, I loved working and living at Cold Spring Harbor, and in 1967 was being considered for a faculty position there by the Carnegie Institution. However, when Jim Watson came to be the director of Cold Spring Harbor in 1968, he let it be known that he planned to convert the lab to an animal virus research center, focusing on tumor viruses such as SV40. Phages were passé. So sadly for me, shortly thereafter, Hershey announced his intention to retire. He was only 60. And Carnegie decided to close the genetics research unit. So I started looking for a job. This was the first time I bumped hard against the gender glass ceiling. I recall interviewing at an institution where I was later informed that although I had a great interview, they already had their token woman faculty member. I think that at that time, this was the point in their careers that most women experienced the greatest difficulty in getting an even break. Well, then I received a rather unusual job offer. Art Weisbach, then at NIH, asked if I would consider joining his department at the Roche Institute of Molecular Biology, a new basic science research institution, which was to be built and funded entirely by the pharmaceutical company Hoffmann La Roche, which, as it turned out, was conveniently located in Nutley, New Jersey, just 12 miles west of New York City, where my husband worked. The arrangement, however, seemed quite unprecedented, and I was a bit dubious. But focusing on research was what I preferred, and I thought I could always move on if the promises of independence were not kept. I agreed to join in 1968, but was to remain at Cold Spring Harbor for another year until temporary labs would be made ready at Roche. I left Cold Spring Harbor in the summer of 1969. At the end of that very same year, Hershey was awarded the Nobel Prize, along with the other two phage group leaders, Max Delbruck and Salva Luria, who happened to be Jerry Hurwitz's brother-in-law. Well, the Roche Institute was the brainchild of John Burns at Hoffmann La Roche and Sidney Udenfriend at the NIH. Burns had convinced the company that such an institute would allow them to transit from a traditional chemically oriented drug company to a modern pharmaceutical company. It didn't hurt that the company was family owned so that decisions could be made quickly and that they were flush with revenue from the sale of Valium. To convince Judenfreund and a large group of individuals to move with him from the NIH, the president of Hoffmann-La Roche signed a charter which promised, among other things, that the institute would be wholly dedicated to long-range research and that scientists would enjoy independence in their choice and pursuit of research problems. The lab at the institute were organized just like a mini NIH, but it is noteworthy that among the initial 25 faculty members and research associates, almost all coming from the NIH, I was the sole female. And that lack of diversity didn't change much over the 18 years that I was there. As with many academic institutions at the time, the Roche Institute was essentially a male bastion. However, I can honestly state that the promises of the company for research freedom that whole time. While at Roche, I initially continued studies on the replication of lambda DNA. One of my first postdocs, Lynn Enquist and I, showed that there are two distinct stages in the replication of lambda DNA. At first, the DNA is replicated as a circle, and later as a rolling circle, which produces the concatenamers that I had described earlier. Most importantly, we discovered that the transition from one stage to the other is mediated by a single strand break, which can be resolved into a rolling circle, either by viral recombination proteins, the red proteins, if the break is in the lagging strand, or by host repair proteins, if the break is on the leading strand. These studies were significant because they were the first to document functional interactions among the pathways of replication, recombination, and repair, pathways that were at the time studied independently in separate stovepipes. We also proposed that such functional interactions must occur during the replication of host DNA, as was later shown to be the case in multiple laboratories. By the early 1970s, improvements in cell culture methods and quantitative assays for animal viruses generated new interest in investigating their properties, and I began looking for an experimental system with expanding horizons. I was attracted to retroviruses because Temin's provirus hypothesis had just been validated by discovery of RT in his and Baltimore's lab and the experiments by Hill and Hillova. Potential analogies with lambda were really intriguing to me. So for perspective again, at this point, restriction enzymes and molecular cloning techniques had just come on the scene. And I was in a perfect position to take advantage of molecular cloning of retroviral DNA in my beloved bacteriophage lambda. To get myself prepared, I did a short sabbatical with Saburo Hanafusa at the Rockefeller in New York City. It was there with his help and that of his generous lab members that I learned how to work with the Rouse sarcoma virus. Now, this was an instant, which was a great advantage to be at the Roche Institute. I didn't have to write a grant or convince the retrovirus community that I was capable of joining their ranks. I just closed down one program of research and later started a new one. Of course, my first goal was to clone and analyze RSV DNA, but that hope was immediately dashed by the recombinant DNA guidelines promulgated at the Asilomar Conference in 1965. As a participant in the conference, the best I could do to move the science along was to help in the certification of so-called safe cloning vectors, one of which was actually constructed by Lynn Enquist, then at the NIH. Enquist's bacteriophage lambda West vector was eventually used all over the world for cloning. Although cloning of viral DNA was prohibited, my postdoc, Bill Clements, and I decided to get some practice by cloning ribosomal genes from chicken cells, from which we could easily make radioactive ribosomal RNA probes. We were able to do these experiments because the Roche Institute had constructed the required BSL-3 facilities for me. And the results were of sufficient interest at the time to be published in a 1976 report in Science. That same year, with the help of Lynn Enquist, McClements and I were able to gain access to a BSL-4 facility at NIH. While there, we cloned RSV DNA from chicken cells using Enquist's lambda vector. It was truly surreal, working in jumpsuits and glove boxes and showering in and out for 2 days, making phage clones from seemingly harmless chicken DNA. Well, after the clones were made, they were sealed in a paint can, the outside of which was sterilized before it could be removed from this facility. And we then took the can back to Roche to be stored in our cold room. We were only able to open it up 3 years later in our BSL-3 facility when the guidelines were relaxed. But then we were ready to go. In 1978, as Andrea said, I was asked to join COGENE, Committee on Genetic Experimentation, which had just been established in response to strong negative reactions against any kind of cloning, especially in Europe and elsewhere, vestiges of which, sad to say, exist to this day. Our first report on risk assessment in 1978 was intended to calm the waters and did manage to prevent outright bans on the technology. In addition to risk assessment, COGENE was also asked to provide laboratory training, especially in places with limited access to recombinant DNA technology. I was very busy in the late 1970s participating in meetings in Italy and the UK, where there was the greatest fear of the new technology. And then after the guidelines were relaxed, I organized a 3-week COGENE lab course in India and then in Hong Kong, and later a lecture course in Nairobi, Kenya. Now, this funky group photo of the class participants at the Hindu University in Varanasi, India, was sent to me recently by one of our course instructors, Barbara Hone. Now, Barbara had invented cosmid cloning and was recruited from her lab in Switzerland to provide that technology in the course. I wonder how many in this audience have ever heard of cosmids. Well, students came from all over Southeast Asia. They are all in the back row in the picture with instructors in the front, together with my old friends from NYU, our host Debbie Verma and Maharani Chakravarti. Now, the late Ahmed Bukhari from Cold Spring Harbor is front center, and Barbara Hone is on his right, and I am on his left. And you can see a seemingly unhappy Joe Sambrook from Cold Spring Harbor at the very left end. As it turned out, many of the protocols developed exactly for this course actually ended up in the cloning manual of Maniatis and Sambrook, as you heard. Back at the Institute during that same period, we were busy harvesting the fruits of our molecular cloning and the newly invented Maxim-Gilbert DNA sequencing. We reported the first identification of an endogenous retrovirus, EV1, in chicken DNA. And from a chicken bred to lack EV1, we cloned the first unoccupied integration site. We also discovered 2 circular forms of RSV in infected cells, having either 1 or 2 LTRs. Most amazingly, our sequence analyses revealed a remarkable similarity between retroviral DNA integration and that of other prokaryotic and eukaryotic transposable elements. Shown at the right in the illustration, the 2 LTRs flanking proviruses are analogous to repeats at the end of the Drosophila retrotransposable elements and the delta sequences that flank the yeast TY elements. And both solo LTRs and deltas are embedded in the DNAs of eukaryotes. Likewise, on the right, or on the left, bacterial DNA transposons are flanked by IS elements, which can also be found isolated in bacterial DNAs. We also noted that all of these elements harbor short sequences of repeats at their termini, and we identified conserved TGCA sequences near or at their ends. And finally, we observed that all were flanked by direct repeats of host DNA, which we surmised at the time were produced at the time of integration. For the retroviruses, these were important landmarks later to be explained in the context of the biochemistry of reverse transcription and DNA integration. Now, there's no way that I can describe the range of activities our lab was engaged in over the next 3 decades. As with others, we rode each new wave of technical advance to new discoveries, or indeed, or is it possible to give credit to all the students, postdocs, and collaborators that contributed to our efforts. But in the next few slides, I'll describe 3 really quite basic contributions from our lab that turned out to be important facilitators and generated a substantial ripple effect in the field. Contributions that illustrate points that I've tried to emphasize with trainees. First, the value of attending seminars in other fields, which many don't seem to have the time to do these days. And the biochemists and geneticists rule that the assay is everything. So the first instance came about during our studies of the endogenous reverse transcription reaction in isolated retrovirus particles. At the time, such studies were performed with particles that had been disrupted with non-ionic detergents such as NP40. But the RT reactions were pretty inefficient. While attending a cell biology seminar, I learned about the fascinating properties of a small protein called melittin, which is the major component of honeybee venom. Now, melittin is a 26 amino acid peptide with an unusual arrangement of mostly hydrophobic residues at positions 1 to 20, here in green, and blue hydrophilic residues at the C terminus. This property makes it highly surface active, and it had been postulated that insertion into membranes produces wedges that traverse the membrane and disrupts its structure. Wow, I thought, this might be just the tool to use on the membranes that surround retrovirus particles. When my postdoc Larry Bruin tried it, he found that treatment with melittin increased the efficiency of endogenous reverse transcription immensely. As listed here, he was able to detect 2 long minus-strand DNA products, one of which included LTR sequences. Furthermore, infectious DNA was also produced in this reaction. The use of melittin was subsequently adopted in many laboratories at the time and turned out to be most useful for studying DNA synthesis in purified HIV particles. I was thrilled to learn when Devin Christensen described his use of melittin in his reconstruction experiments. And as Larry Boone had shown with RSV particles some 30 years ago, infectious viral DNA is produced in his system. Well, with our RSV permeabilized particles, we also showed that the viral RT can perform strand displacement synthesis, an activity that's really critical for formation of the LTRs. Now, this activity was especially robust in the RT of RSV, as we could detect in the EM, structures with intact minus strands and discontinuous tailed plus strands formed by the activity. I hope that all of you had the correct answer for number 3 down in the retrovirus trivia crossword. My next 2 examples followed emergence of the HIV pandemic. which focused everyone's attention on viral enzymes as potential drug targets. One of our most cited papers resulted from a collaboration with investigators at NIH in which we demonstrated that the HIV protease is essential for virus reproduction. This was no surprise, really, as the principle had been established earlier with RSV, but the data had to be obtained. Now, at the time, Hoffman-LaRoche chemists had just perfected methods to make small peptides synthetically. It occurred to us that these reagents could be used to determine the specificities for processing retroviral GAG and GAG polyproteins. So, with the help of these chemists, we developed the first peptide assay for retroviral proteases. Now, using a variety of synthetic peptides that they made for us, we determined that for HIV, the critical length for binding and cutting was 7 amino acids, that residues at the center were especially critical. If you substituted other amino acids in these residues, you produced peptide inhibitors. And then, in collaboration with John Leese and Alex Lodauer, we obtained the first molecular model of HIV protease with a bound peptide. Now, the peptide assay, together with computer-assisted drug design, paved the way for identification and development of non-peptide protease inhibitors, with the Roche drug Sequenavir the first to gain FDA approval. In 1987, I received a call from a headhunter asking if I would consider moving to the Fox Chase Cancer Center as director of the Institute for Cancer Research. It was a good time for me. Our children were grown and out of the nest, and I began to have doubts about how long Hoffman-LaRoche would be willing to support the Roche Institute in Nutley. As it turned out, not much longer, as the company decided to close it down some 5 years after I left. The Institute for Cancer Research, much like the Roche Institute, was fairly small but prestigious and devoted solely to research. It also had an excellent history of supporting women, with several well-known female researchers on its faculty at the time. So in 1987, I moved my laboratory to Fox Chase with the help of my colleague, Rich Katz, who remained a research partner for many years. Now, much of the work in the early days at Fox Chase was focused on the retroviral integrase. At that time, everyone was trying to discover specificity for its known endonuclease activity. To ask this question, we and others had been using plasmid DNAs containing LTRs, with only marginal success. Efficient methods for oligonucleotide synthesis were just being developed. And thinking about our experience with protease, it occurred to me that we might have better success if we used short oligonucleotide duplexes as substrates, in a sense, to focus the attention of the enzyme to where we believed it must act. So together with Jonathan Leese, we developed an assay that presented the enzyme with short duplex oligos that mimic the tips of the LTRs, shown in B at the right. Using gel electrophoresis, we found that the enzyme not only processed the oligos at the correct site, adjacent to the conserved CA, but also joined them to other oligos in the reaction. It was an important breakthrough, presented first at a poster at this Cold Spring Harbor meeting and rapidly adopted in the field. Using this assay in 1990, our lab and Bob Craigies reported that the RSV and the MLV purified integrase proteins were both sufficient and necessary for integrative recombination. Later work by us and others used oligos to detect concerted integration for 2 viral DNAs. As for the ripple effect, the oligo assay, or derivatives thereof, has been used by everyone in the field and most importantly formed the basis at Merck for identification of the first antiviral inhibitor of the HIV joining reaction, raltegravir. Now, with nucleic acid sequencing becoming more routine and bioinformatics more mature, we then identified the critical DDE motif common to integrase, retroelements, and bacterial insertion sequences. We showed that integrase can turn over catalytically. It's a real enzyme which functions as a multimer. And then together with Isaac Wang, using pre-steady state kinetics and atomic force field microscopy, data shown on the right, we obtained the first physical evidence that integrase functions as a tetramer. X-ray crystallography, in collaboration with Alex Lodauer's group, produced one of the first structures of the integrase catalytic core and revealed the similarity of its active site and mental cofactor binding to RNase H, shown in the middle at the top. And we also reported one of the first structures of the catalytic core domain with a bound inhibitor at the bottom. Sadly, despite subjecting what seemed like gram quantities of full-length RSV integrase to crystallization trials, we were never able to obtain the Holy Grail. That honor was reserved for Hideki Ihara and Dwayne Grangenet in 2006. But we did have some success using small angle X-ray scattering together with chemical cross-linking and molecular modeling in 2011. And in 2013, these studies revealed that in the absence of DNA, HIV integrase forms two types of dimers in solution, shown in A. At the left, a dimer based on interactions between the catalytic core domains, and at the right, a dimer in which the core domain of one monomer interacts primarily with the N-terminal domain of the other. Now, both interactions are critical in the conserved intosome core, which is a dimer of dimer arrangements in the HIV intosomes and others that have been looked at. Several groups have demonstrated that the core dimer interface can be a target for antiviral drug development. The potential of the NT core dimer as a target for drug development is currently being tested independently by my former colleague, Mark Andrake. In 2008, I took the opportunity to dispense with administrative duties at Fox Chase. 20 plus years as director seemed like enough for anybody. I was ready to focus solely on science. At that time, genome sequencing was taking off, and the era of big data had begun in earnest. At the invitation of Arnie Levine, I decided to take the second sabbatical of my career at the Institution for Advanced Studies in Princeton. Now, Levine was head of the Simon Center for Systems Biology, and his goal was to promote collaborations among biologists and computer experts to take advantage of the coming plethora of big data. My collaborator, shown here, Vladimir Belyi, was a young physicist with expertise in data mining, who happens now to be a VP at Goldman Sachs. We first planned to study endogenous retroviruses in the genomes of various vertebrate species. Instead, we ended up discovering fossils of viruses for which there were no previous archaeological records. At one of the tea time breaks, which are traditional at the Institute, I mentioned to Vlad that I had heard reports of sequences of other plus-strand RNA viruses, such as flaviviruses and picornaviruses, in the germline of insects and plants. This was curious because these RNA viruses had no DNA intermediates in their genome replication. Well, we wondered, what about vertebrates? And we decided to have a look. The strategy that Vlad developed was to scan all publicly available vertebrate genome assemblies. There were only 48 at the time, for sequences encoding proteins related to those in single-stranded RNA viral genomes, other than retroviruses, for which about 6,000 were then available. Well, the results were nothing short of amazing. We identified 80 high-confidence germline integrations of RNA virus sequences in 19 of the 48 vertebrate species for which DNA was available at the time. In all cases, they appeared to be copies of mRNAs from highly expressed viral nucleocapsid or polymerase genes, and nearly all were related to only 2 of the known single-stranded RNA virus families, the Bornaviruses and the filovirus, Ebola and Marburg virus. By phylogenetic comparisons, we found that most of these sequences had been inserted in the genomes of vertebrate ancestors more than 40 million years ago. It's interesting that this was about the time of mammalian evolutionary radiation. Now, one exception was the squirrel, which seems to have acquired its insertion more recently. I'll come back to this a little bit later. Now, several of these viral sequences appear to have been inserted via line-like elements. They had poly A sequences at one end, and they were flanked by host sequence duplications. Most significantly, some of the viral open reading frames were entirely conserved over these millions of years, and the sequences were expressed in certain tissues, suggesting selection for some important function, the need of which persisted over a long period of evolutionary time. Now, what is about these filoviruses and the Bornaviruses that could account for such findings? Well, we know that both viruses are fairly lethal pathogens for the species that they infect. For example, the case fatality rate for Ebola in humans is 50 to 90%. And for natural infection of Bornaviruses in horses, the case fatality rate exceeds 80%. So we speculated that the conservation in the genomes of certain vertebrate hosts might be related to their pathogenicity. We noted at the time that there was indeed a suggestive correlation between the absence of Bornavirus fossils among the hosts tested and their susceptibility to infection by present-day Bornaviruses. Vertebrate species that had germline integrations, such as primates and rodents, shown at the top of this table, were not natural hosts. And we speculated that expression of the ancient viral genes might provide some intrinsic genetic immunity, for example, by a dominant negative interference of slightly different proteins during virus reproduction. In support of this notion, it was later shown in Tomonaga's lab that expression of one of the squirrel endogenous Bornavirus sequences block the reproduction of a mammalian Bornavirus in human cells and culture via inhibition of the viral polymerase. Belli and I went on to identify fossils of ancient small DNA viruses in vertebrate genomes as illustrated at the right. Now, these DNA integrations were found to have occurred over a vast expanse of evolutionary time and are easier to rationalize from the known genomic replication mechanisms of their currently circulating relatives. If I had another research life to live, I certainly would have investigated the significance of our findings further. But no worries, others joined the fray very quickly. We seem to have been at the vanguard of a whole new field called Paleovirology. After my sabbatical, I returned to Fox Chase to spend my last decade with the retrovirus. And in 2016, I decided that it was time for me to step aside from the lab and make room for the next generation who seemed well positioned to surf the next wave of technology development, of which we have seen so many exciting examples at this meeting. But I was no means finished. Indeed, as an emerita professor, I now enjoy the leisure of a scholar. And as I look back at my experiences with retroviruses over the years, I was inspired by how much our community has learned over these last decades and in so many fields, genetics, evolution, and medicine. It was a story I thought should be told for everyone, students, other scientists, and even the lay public. So I wrote a book directed at this general audience, Discovering Retrovirus, Beacons in Our Biosphere. It was a challenging but rewarding undertaking, allowing me to review some of the earliest literature in the field, some of it truly extraordinary. The book is short and the contents are shown here. It tells some short stories about key people and has a number of simple, I think, very nice illustrations. I think even some retrovirologists have enjoyed reading it, and I hope it'll be recommended to students. My second most important scholarly undertaking has been the 5th revision of the ASM textbook Principles of Virology, with co-authors Jane Flint from Princeton, Vincent Racaniello from Columbia, Theodora Hasianazzo from Rockefeller, and Glenn Roll from Fox Chase. Preparing this textbook has truly been a labor of love from the start of the first edition some 25 years ago, but it gets better with every edition, and the latest should be available in July for courses that start in September. Well, I've come to the end of my still counting. As I look back at this cascade of discoveries and technical advances, it's also clear that the situation for women in science has advanced over these last few decades, but we're still not there yet. There is more yet to be done, not only in the US, to realize the potential of the female members of our scientific community and to properly acknowledge their achievements. In any case, I hope all of you in our virtual audience have enjoyed hearing this selective account of my personal journey through the decades. But I do miss the ability to interact with a live audience and sincerely hope to hear comments or questions for you now or in the future. And I want to end by saying thanks very much to Ron and Zandria and the other people and the people in the the Cold Spring Harbor crew for this lovely opportunity.
Thank you so much, Anne. I really enjoyed your talk and hearing about your history in the field. So it sounds like if you told us what you would work on outside of retroviruses, but if you were to continue your retrovirology work, what area or what field of retrovirology do you think you would continue to work on now?
Oh, gee. I'm not sure. I'm really very, very interested in what's going on inside a virus particle as it enters a cell. And I think there's an awful lot of nice work that's going to be done there. But we do really need to get some more technology improvements, I think. But that's where I think I'd like to go.
Does anybody on the panel have any questions for Anne? Well, we have some questions.
Yeah.
Oh, there you are, John.
Inspiring talk, Anne. Thank you very much. So what do you think about the mechanism of recombination?
Mechanism of recombination? What are you talking about?
Just
whose recombination?
we both dined out on this for several years a while ago, as you may recall.
Yes, yes.
Are you going to concede?
Oh, you're talking about RNA versus DNA.
I'm talking about template switching versus
Yes.
plus strand invasion, I think it was.
Yes. Well, I concede.
Very good, thank you. I can't help teasing you a little bit about that.
So we have a couple of questions, I think, from the audience. A lot of people are saying thank you. Louisa from the UK is asking, is there anything DNA, RNA, retrovirus related you haven't worked on?
Yes. I don't know. Probably not.
Fair enough. Someone anonymous is asking for your comments on salary equity in science.
Salary equity in science.
I'm assuming male to female scientist salary equity.
You know, I personally, I don't really know very much about that. I know about it in my institution, and there is no problem there. It's quite equal there. Depends on your accomplishments and your contributions to the institution. I don't know how it is in other countries, but in our institution, I think it's quite equal.
And let me ask a question. Looking back, which of the many different things you've done, it's always been science, but your role has changed over the years. Which one did you enjoy the most?
I don't know. I think with the early times of studying retroviruses was really the most fun, I think. When I switched into the field, because it was completely new to me, and it was very invigorating to go into animal systems, and that was the most fun for me.
So another question from CV Dash in the audience is, what is your advice on how to balance basic science with so-called fundable science?
You know, I don't know. It's a really tough time these years now. These last few years have been very, very tough. I was always inspired by doing what I thought was fun, and I got away with that. But I know that's not easy these days, and it's not easy to be a scientist all by yourself these days. It's big groups. There aren't many papers with 1 or 2 people on them as there were then. So it's a different world, and I don't know how to give advice that way. It's really tough. Yeah. It was time for me to leave.
So Alice Telesnitsky is asking, any advice on how to balance leadership and research roles?
Leadership and research work, yes. Well, I think I was very lucky because I had recruited very early on a group of people who stayed with me for a long time and helped me continue the lab while I did a lot of this administrative stuff. And that was a big, big help. So I think if you can get some lieutenants or some other coworkers who are interested to stick with you for a long time, that's the easiest way to get it done.
Were you, did you believe in delegating to people in the lab or around you in the department?
Delegating in what way?
In like leadership responsibilities.
Oh, definitely. Yes, definitely. Yeah. Yeah.
Paul Spearman is asking, what book are you going to write next?
Haven't decided yet.
Namita Kumari says, thank you for the lovely talk, and says, I have used Cosmid for genomic library cloning. Thanks again for all the basic tools. Someone is asking, would you switch to work on coronavirus if you had a lab at this time?
Coronavirus? I don't know. Maybe.
Okay.
Maybe.
Okay. Any advice that you would give to young scientists right now, starting out in their career?
I think to succeed, you have to really love what you're doing and find what it is that drives you beyond any reasonable stopper, and just go for it. I don't know what else to say, because I was always driven by my interest and wanting to know. I don't know any other thing that will help you be successful.
Any advice on overcoming potential roadblocks, particularly for budding female scientists who are trying to climb the ladder?
I never accepted that there was any problem with my being a female. I just went for it. I just ignored the problems, and there are some. There always are some, but I just I didn't let it get in my way.
Okay, I think that is it.
Good.
Thank you so much, Anne. I think you've inspired a lot of people in the audience, and I think a lot of people enjoyed it. So thank you for being here.
All right, thanks.
So before we sign off for good, Ron and I want to thank all of you for showing up. I was worried that no one would actually attend, in this virtual space. So I'm glad we had so many participants. Thank you for everyone who submitted an abstract and presented a talk or a poster. Thank you for all the chairs who ran the sessions pretty flawlessly. And I think, the fact that this meeting ran pretty well is due to the Cold Spring Harbor staff, particularly the AV team. So I'd like to give a huge applause to them, as well as Katie Carr and Mary Smith, who sent us lots of emails and told us what we were supposed to be doing and when. And of course, David Stewart, who is sort of running the big picture and make sure that we're able to do this every year. I don't know, Ron, if you have anything to add?
Well, I want to thank Sandria. Sandria's been great to work with, and everything that worked well in terms of the organization is because of her attention to detail and being very thoughtful in her choices. So thank you, Sandria. And last thing I want to say is this will forever be known as the meeting that proved that coronaviruses will never take down retroviruses. So thank you all very much, and we'll see you next year.
Yeah.
Yes.
Thank you, Anne.
Okay.
Yeah.
You're welcome. Thank you.
Thank you, Ron, for being such a great mentor.
Elf. All right. Bye. I think you owe me a beer now.
Definitely, at least one.
Okay. Bye-bye, everybody.
Bye.